Um, Good morning, Dr. Tapan. Good morning, Padmini Madam.
Good morning, Dr. Sundaram.
Morning.
Good morning also, Dr. Tapan Mondal. Nice to see you. Your topic is so excited. So even though I have some other assignment, I will attend for some time, Dr. Sundaram.
Thank you very much, ma'am.
You have also
It is so
joined
nice.
online cutoff. Right.
Good morning, sir.
Good morning, Padma ma'am.
Good morning, Padmini madam.
Good morning. Good morning.
Good morning, Tapan sir.
Good morning.
Mark kochi ebe koichi senior ko dekhantu kono.
Actually, today morning suddenly the weather is
meeting
really
ଅଛି
become
ଆଜି 3-4ଟା meeting ଅଛି
ଆପଣ ଡାକିଥିଲେ ବୋଲି ମୁଁ ଆସିଲି।
Morning, Niraja madam. Morning.
Good morning, Dr. Mondal.
Yes.
Sir, we're not able to see your face, sir.
Uh,
I think you need to adjust your camera accordingly. Yeah, now you're visible, sir.
okay. Actually, I have to tilt my computer then. Fine. Actually, from today morning, the weather has become very bad in Delhi. Very dark, rainy. There maybe it should not be any load shedding or power cut.
So shall we wait just for another 2-3 minutes so that more number of people will join? Padma Madam, shall we start? Hello?
Yes, sir. Yeah, very good morning, sir. Now, I think Dr. Padmavati will take over, sir. She'll give her the introduction. I think 36 participants are there online, sir. Should we wait for 5 more minutes?
I think we can start it, ma'am. They'll join in due course, sir.
Okay.
Some are present in the committee room also.
Okay, sir. Okay, sir. Dr. C.H. Padmavati, are you online?
Yes.
Okay, madam, carry on.
Okay. Good morning to all of you. I welcome you all to today's webinar on Oryza coarctata, a potential source of stress tolerance genes. At the outset, I extend my warm welcome to today's chief guest, Dr. Tapan Kumar Mandalji, principal scientist working at ICAR National Institute of Plant Biotechnology at New Delhi. Sir, on behalf of the Indian Institute of Rice Research and also on behalf of Society for the Advancement of Rice Research, I extend a warm welcome to you for today's talk, sir, and thank you for accepting our invitation to deliver this talk. Thank you. And I also welcome our beloved director, Dr. R.M. Sundaram, sir, who is also the president of SAR. I welcome you to this webinar, sir. I welcome Dr. Director NRRI, Dr. Padmini Swain ma'am, who is online. I welcome you, ma'am, for this webinar. And I also welcome all the scientists, rice fraternity, and also the members of SAR and scientists from various institutes I could see from IARR, NRRI, IRI, and also all those who have joined us today for this talk. I welcome you all. Thank you.
Thank you very much, ma'am. It's indeed a matter of great happiness for all of us that we have an eminent scientist and he's also an excellent human being, my friend Dr. Tapan Kumar Mandal. He's going to speak on Oryza coarctata, a potential source for stress tolerance genes. As you all may be aware, Dr. Mondal has done pioneering work not only related to Oryza coarctata, but also in general about genomic resources involved in wild rice. And sir, on behalf of ICAR Indian Institute of Rice Research, ICAR National Rice Research Institute, and also on behalf of Society for Advancement of Rice Research, I extend a warm welcome to you and also to all the participants. And we look forward to hear from you about the exciting recent developments in your research group and also the status in general about exploitation of wild rice genomic resources. Sir, you're most welcome, sir. I now request Dr. Girish to introduce the speaker, Dr. Tapan Kumar Mandal.
Yeah, thank you, sir. A very big good morning, respected Director Dr. R.M. Sundaram, sir, Chairman Seminar Committee, colleagues from the different institutions, students and friends. It's indeed my pleasure to introduce today's speaker, the eminent scientist, Dr. Tapan Kumar Mandal, sir, from ICAR, National Institute of Plant Biotechnology, New Delhi. So Dr. Mondal did his BSc and MSc from Assam Agriculture University, Jorat. He has obtained PhD from Institute of Himalaya Bioresource Technology, CSIR, Himachal Pradesh, India. After that, he served as a research scientist on Tata Title Limited, Munnar, Kerala, and later as assistant professor at Agriculture University, West Bengal. After that, he has joined as a senior scientist at Indian Council of Agriculture Research, Ministry of Agriculture, New Delhi, and presently working as a principal scientist at ICAR, National Institute of Plant Biotechnology, New Delhi, on salinity and utilization of wild species of rice. He did his postdoctoral training at University of California, Riverside, USA, and University of Urbana, Illinois, USA. Dr. Mandal, sir, is the recipient of several scholarships and fellowships of Professional Societies of India, including NASI Fellowship, NASI from Allahabad. He mentored farming community of Sundarban, West Bengal, for which he received a prestigious Genome Saver Award for conserving a salinity tolerance indigenous rice genotype. He has also visited several countries, written 2 books on plant biotechnology of Springer publication. He has published more than 85 scientific research papers and guided 11 PhD students, including 1 from Nigeria and several from MSc students. Today, indeed, it is a great opportunity for all of us to listen the talk on Oryza coarctata, a potential source of stress tolerance genes from Dr. Mondal, sir. I, on behalf of IIRR, SCSR, and on my personal behalf, cordially invite you once again for today's lecture, sir. Thank you, sir.
Thank you. Good morning to all. My slides are visible, I guess.
Yes, sir. Yes, sir. It is visible, sir.
Okay. So, respected colleague, Director IIRR, Dr. Sundaram, and senior colleague from the IIRR, Dr. Niraja Madam, and also I could see Dr. Padmini Madam from NRI Cuttack colleagues. So, on the outset, I'd like to thank to the Director, Dr. Sundaram, to give me an opportunity to present my work. Basically, I'll be presenting my work on the Oryza coarctata. And secondly, I would also like to apologize if anybody is listening my lecture for more than 3 times, because many of the things will be common, because this is my work which I am going to present from 2015 in 45 minutes. So I'll try to explain the recent development of the Oryza coarctata. But I would like to say that in my lab, I work on 2 different crops. One is the rice and wild rice, another is the tea. In rice, I work on the salinity and deep water rice in Oryza coarctata as well as the Oryza glaberrima. And in deep water rice, I work with the collaboration with the Assam Agriculture University. But today, I will be restricted my talk on the Oryza coarctata. Therefore, my title of the today's talk is Oryza coarctata, a potential source of stress tolerance genes. As many may know, but many are already knowing about this plant. This is a species among the 27 species of the genus Oryza. It is the most primitive species, belongs to the tertiary gene pool of the genus Oryza, and it can flower up to the maximum salinity EC of 40 to 45. This is not a very easy crop, unlike the rice, which belongs to the A genome and primary gene pool. There is a lot of difficulties with this crop, like it is a perennial, mainly propagated by vegetative propagation. There is no known economic value on this particular species. Also, botanically speaking, it does not make a cross with any of the species of the genus Oryza. Chromosomes are very sticky, small, and most importantly, no institute is having the germplasm collection of this particular species, like including the NDPGR as well as International Rice Research Institute, Philippines. They have very limited collection of this germplasm. So work related to population genetics, etc., is not able to do this crop. And this crop basically grown in the coastal region. Then people often ask me that why you are working on this crop. There is a 2 main reason for this. One is that, very importantly, this is significantly tolerant to the salinity and submergence among all the 27 species from the genus of Oryza. And secondly, the person or people who knows this crop more than 40 to 50 years they have informed that there is no known pest and disease reported for this crop, which means that this particular, not crop, this particular species is tolerant to various kind of biotic and abiotic stress. And I am handling it from the 2013, and I have not seen any pest and disease on this particular crop, although it is known for salinity tolerance. So that's the reason. And the most important thing is that this particular species originated in the Indian subcontinent. So it is our responsibility, some of us, to work on this crop. It does not available in any other place in the world. I'll show you. So this was the main reason for which we got interest to work on this particular crop, and it was known to be tetraploid and with a KKNL genome. I have written it in the italics because end of the lecture we will find that there is some changes on this. And as I told that it is also acid sulfate tolerant because it grows in the coastal region. And when we started this work, 2014-15, there was a very limited genomic resource available on this crop, on this particular species. Only 72 nucleotide sequence was there in the NCBI and 400 ESTs, and only 4 RNA sequencing was there. So, but I must recognize the previous work on this crop. There are 2 institutes in India who was working. One is the MS Swaminathan Research Foundation, Chennai. Professor Swaminathan, he himself used to work on this crop. And the focus was they have cloned some of the SOS pathway gene. SOS is a major salinity responsive pathway known to the plant, including the rice. And say they have cloned a couple of genes from these, including the sodium transporter NHX gene. And Bose Institute, Professor Lairi Majumdar did an extensive work on the pinital biosynthesis pathway. All of his research collectively established the fact that pinital is a pivotal role or major role for giving a salinity tolerance of this crop. But not much work on its regeneration and other thing was reported except these 2 institutes. There was a massive attempt to make this cross from this Oryza coarctata with the cultivated rice, but with a very limited success. Out of 20,000 cross in IRRI, there was a successful cases of crossing. Even in Bangladesh, University of Dhaka made some cross with a limited success. So therefore, like any orphan crop, the then director of NIPB, Dr. Tilakar Sharma, who is the present DGCS, and myself, we decided to go for decoding the genome, because that in a single shot will enlighten many of the things. It will generate the genomic resources, what we can use later on. So we decided to go for whole genome sequencing of this crop, of this plant, and we have collected the material from the Sundarban. Let's have a look on the availability. As I said, it has been reported extensively and known that it is available only in the coastal region from the Myanmar, the Bangladesh, and in the Indian coastal region, this 7,500 kilometers, including up to the Karachi. When we try to get more information, we are making attempt to collect different accession from different parts of the India. We found that it is also not available in all the entire coastal region. It is high association in the mangrove ecosystem, particularly with the Avicennia alba and other Avicennia species. So wherever the mangrove ecosystems are there, like Andaman, Sundarban, Orissa, Bhitarkanika, then in Kutch area of Gujarat, even in Karachi. So there these plants are available. As I shown in the first figure, that these plants grows below the mangrove trees and make a bed on that, because it grows primarily with the roots vegetatively. So it makes a mat below the mangrove trees, and it is grazed by the cows and buffaloes. That's all. And one of the interesting fact and our information is that if you see the area or the coastal region where it is available, the EC of the coastal region varies for Oryza coarctata from 27 to the 67 EC, and this is one of the report. I have taken this paper from there, and also it also available from the coastal region to 2 to 3 kilometers inside the land, and which also consists of the brackish water, and the EC of the brackish water started from the 4 and above. So what I want to say, if you look back to the soil EC on which the Oryza coarctata grows, it varies from the in the natural condition, varies from the 4 EC to the up to the 60 EC, but it comfortably go up to the 45 EC. In a recent paper is shown. So what our hypothesis is that it goes in the different soil EC in a very varying soil EC. So there may be some variation among the adaptability of the salinity on the Oryza coarctata germplasm, but which is yet to be explored. We are trying to do that. And if we get the variation on that, then the research can be more advanced if we get some kind of variation on the salinity response. But as on today, my work is limited only from the Sundarban collection. So considering that, my outline of the talk is that I'll be talking on the genome assembly of this crop, which we have done at NIPB, and then cytogenetic studies, and then LCMA studies, and then finally RNA-seq studies. We have also the failure theory that with the help of the genetics department of IRI, we are trying to make a cross for last 4 years, but we did not get any success on that. So, sorry. So when we start the work, we first look for the genome size of this crop. Then we did not find any of the reference. Only one paper was there from the Rode wing group. They reported that the genome size was 1,283, although in a asterisk they put it that this was the assumption based on the in silico analysis. So we thought to do our own genome size determination. And before that, I'll just have a brief quick outline about the genome sequencing work of India. So far, there are 13 crops which has been sequenced by purely funded by the money from the India. There is no foreign collaboration here. And out of that, the yellow marks are done from the NIPB, either directly NIPB has done or with association or with the other institute. Among this yellow, these two greens are from my own lab, that is Oryza coarctata and on the Indian tea. So I'll be talking about the Oryza coarctata only. These are the picture of those crop, and the circle represent the genome size. What I want to say that the different crop is having different genome size, and the genome sequencing work depend on the genome size, because genome size determine the time required for genome sequencing and the cost and energy, everything. And you can see that Oryza coarctata coming in between with the 665 MB. And so we went for the genome size determination. To our surprise, we have found out that through flow cytometry, taking PCIMM, SATIVM as a internal standard, 665 MB against the previous report, which was anyway an assumption 1,283 MB. So with this, we start the genome sequencing work. Those who are not very familiar with the genome sequencing, quickly I will be explaining this slide that will help you to understand my work. In any genome sequencing work, we first break the DNA into the small pieces, and these small pieces are then being sequenced. And then second part of the genome sequence is that how efficiently we can align or we can attach those small sequences to make the big sequences as equivalent to as individual chromosome. So part one is the break the DNA, make the sequence, and second part B is that to add those small sequence to make it big as the size comes close to the individual chromosome. So in bioinformatics language, we call pseudomolecules, but basically this is equivalent to the chromosome. But the merit remains here in two things, that when we break the DNA and do the sequence, we use different length of the sequence, because in varying length, smaller to medium to bigger, it is easy to align them in their same position. Second point on which the cost of the genome sequencing depends is the generation of the data. That is, with the more the data, it is better, it's near accurate. We get the good alignment of the small sequence to make it the bigger. So, which we call in technical terms is the depth. Depth is nothing but if you put the finger on any point of chromosome, so we see that how many layers in there, particularly in that point, the sequence are there. The more the layers, the more the depth, higher the depth, and more confidence we get, because in the same point, if we get the same nucleotide sequence A or 200 sequence, then we can confidently say that this particular point is having A in the genome. Otherwise, during the sequencing, a lot of error occurs, and then later on we get confused. So the depth of the genome is important, and the different type of the platform or different length of the sequence we used to add the small sequence to make it the pseudomolecule is the important. So this is common for any genome sequencing project. So this is the outline and which we determined at that point of time in 2017. So first what we did, we took the Oryza coarctata genome and we go for the Illumina short read, and the length of the read is 151 base pair in all these. So in Illumina pair and read, we generated the 124 GB data, and with the help of software, we try to put them and align them to make them bigger. So when we do this, and then we got the around 1 lakh contigs. But ultimately, our aim is to reach the 12 basic chromosomes. So this contig, if we are efficient, should reach to the 12th chromosome. So to reduce this contig, then we use the Illumina met pair data of varying length, 2 kb, 4 kb, 6 kb, 8 kb, add on to this backbone. And you can see the contig has reduced from the 1 lakh to the 91,000 contig. Here it is not enough. As I said that if we make a perfect genome sequencing of the Oryza coarctata, we should get the 12 finally pseudomolecule or 12 large contig. So to reduce further, we use the Oxford nanopore during 2017. And then you see that when we put this Oxford nanopore data, Oxford nanopore is the next generation sequencing. The important point of the Oxford nanopore is that it produces very long read. And in our case, you will see that we got as high as 660 kb long read of the Oxford nanopore. And it is enzymatic free, so that's why it is possible to make the larger sequencing fragment. So when you put this Oxford nanopore in the backbone of this Illumina met pair, the contig further reduced and it become larger. When the contig become larger, we call them as a scaffold. And then the number of scaffold has reduced to 58,362. And then we submitted this to the NCBI and made it open. And we published this paper with 58,362 scaffold. But I must confess here, this is not very good genome sequencing because this is a draft genome sequencing. Ultimately, the target of the genome sequencing project is to make the genome sequencing in the chromosomal level so that you can assign all the gene marker as per their position in the chromosome. During this genome sequencing work, so we also took the advantage of the Oxford nanopore read, and then we have made the reference-based chloroplast genome, and then we finally also got the chloroplast genome sequence of this crop. Similarly, this was the reference-based because we have taken the help of the 11 different Oryza species. We also took the de novo approach and did the mitochondrial genome. So in a single genome sequencing work, we have done the nuclear genome, chloroplast, and mitochondrial genome. And so that we have published, but then our aim was to achieve the chromosome level sequencing. So now we are working on that. So what we did on that, we have added the back library. So we had the back library. So we have back library sequence around 2 lakhs. And then we added into that assembled, which are submitted to the NCBI. And you can see the result. Initially, it was 58,000 plus. Now the scaffold has reduced to the super scaffold, much larger size in the 12,000. So it is still not sufficient. We are making attempt to make it 12 pseudomolecule. So now recently the work is going on. We have the high-C data, and also we have massive RNA-seq data of this plant because our aim is to develop the chromosomal level genome sequencing and reduce this super scaffold to the 12 pseudomolecule. So this was the present status of the genome sequencing of the Oryza coarctata. It is still going. We'll be coming to the version 2 of the chromosome level genome sequencing of Oryza coarctata shortly. These are the stat of the genome sequencing. So as I told you beginning that the depth matter. So our present depth is 250x, including the parent and met pair and nanopore data. And in this below diagram, one important point is that we were the this is the declaration from the Oxford nanopore. In India, it was the first time that Oxford nanopore in that time with the mini-ion chemistry has been used for any higher eukaryotes. And it was given in the Oxford nanopore for a long time in their website. Although the error rate in that time was 13%, now they have an improved chemistry on which the error rate has been reduced in the Oxford nanopore. So we got the maximum wavelength maximum sequence length with the Oxford nanopore near 700 kb, and that helps us to hook up the chloroplast and mitochondrial genome from this data. Because of the larger sequencing length, it is more than the size of the chloroplast and mitochondria. So these are the basic stat. Important stat here is that estimated genome size, what we got through the flow cytometer is the 665. Our depth was 250x, and we could cover 570 base pairs. So this much is still left. We have a gap of this, which we are trying to improve this genome sequencing using the back library as well as the high-C data. And so we'll be developing the chromosome level. At present, our genome sequencing, whatever we got is a draft genome, but we are trying to achieve the platinum standard genome sequencing of the Oryza coarctata. Repetitive region is 20%, and very interestingly is that out of 27 species, whatever has been done genome sequencing, this is the lowest repeat region which is present here. But the biological significance is not known or yet to be known. And gene model, we have established 33,000 plus. So when you do the genome sequencing, very quickly, people ask that how good your genome sequencing. There are two parameters. One is the sigma, another is the BASCO. If the reading is more than 90%, it is considered to be a good one. So our BASCO sigma reading was 92. After normalization with the RISE genome, it has increased, but original stat was about 92.34%, and BASCO reading was 97.08%. Although it was a draft genome, but the genome assembly was, or completeness was good as shown by the sigma and the BASCO. We also did some standard analysis, which is there in all any genome sequencing projects. We have more than 2 lakhs SSR of varying length, but it is assigned to the scaffold level now, super scaffold level, because we have not the chromosome level sequence now. We have also find out some around 5,000 transcription factor of 56 different family, with beta helix is the larger family. And then the interesting point is that out of 33,000 plus gene, we have find out 2,260 gene which has not any hit with the NR database, which means that these are the novel. They don't have any geoterm, neither has any IPS match, protein sequence match. So we are interested to look what is this novel gene that is present. With the limited resource, we have identified the 11 gene that we are, which is highly upregulated under the salinity stress. It is an uncharacterized novel gene. We are trying to understand this through the transgenic approach, and 2 of them I'll show you now. This is another, as I told, the repeat region is present here, the 20%, and these are the different transposable element and other repeat region which is present in the coarctata genome. And these are the non-coding sequences which has been dig out from the genome, although we have much larger microRNA and other non-coding RNA from here. So one of the interesting points we wanted to know about is relationship between among the different species of the Oryza genus. So far, whatever genome sequence is available, we have taken there on the basis of 1:1 single copy orthologous gene, a method which is widely used to study the genetic relationship with this. We found that the Bachiantha is quite close to the coarctata, although Bachiantha is the FF genome. But people, those who knows the Bachiantha, they say that they looks morphologically quite similar than the other species. So it may be that it has some relation with the Bachiantha, which is not known presently. We also did the synthetic study with some crop. Here we have taken 2 model crop, Arabidopsis thaliana and rice. And apart from that, we have also taken 2 halophytes, which are present. Both of them are dicot, and genome sequence was available. So we have done the synteny, but the interesting fact is that we have found 8 scaffold, which represent 2.19% of the Oryza coarctata genome, does not have any synteny with the Oryza sativa. It means that these sequences are not present in the Oryza sativa, but present in the Oryza coarctata. We are looking into this, and as I told you that there are 2,000 plus genes which has no hit in the inner database. They are mostly located from this scaffold. So we are working on this, try to understand, and we guess that these are the important region of the coarctata genome which may be responsible for the high salinity region. These are the chloroplast genome which we have done, and after doing the chloroplast genome through the bioinformatics, we have done the wet lab analysis. We have designed the primer from the different region and amplify and then sequence and then blast it again the chloroplast genome, and we find that it has the exact match that gives the confidence to us that what we got through the bioinformatics has the real result, because we have got through the wet lab. In chloroplast genome, it has a 82 number of gene, and other standard analysis we did. Similar, this is the mitochondrial genome, which is 490 kb long, and it has 91 gene, and these are the different strat of that. And then this paper was published with the draft genome before the back, and it does not have any back and high C data, which you are doing now. So this was all about the genome sequencing of the Oryza coarctata. Then the utility point of view, that as I told that we have identified the 11, we have chosen the 11 gene to work. These are the criteria for that. The novel and unique gene was identified. Length was 600 to 2 kb because larger than that will be difficult to clone right now. And they have a very low or no protein sequence similarity, and the relative expression under salinity through qPCR of this 11 was very high. So to do any qPCR analysis, you need to standardize the reference genome. And in coarctata, there was no reference genome was fixed. So one of my students, he established that actin and elongation factor 4 alpha are the most suited reference genome under different abiotic stress of the Oryza coarctata. So all the work we do the genome gene expression, taking these two as a reference genome. So as I told you, these two genome gene, we have chosen this. They're 1 hour and 6 hour. This is their expression level under the salinity. So we have taken them, we have cloned to the vector, and we have made the Arabidopsis, transgenic Arabidopsis, and we have found the fantastic phenotype. They are highly tolerant against the salinity. And the recent result, they told this is the T3 Arabidopsis plant. And we have also done the localization study, and you find the top panel is the control. Second panel is the 1330 gene. And it has been found that it is being expressed in the nucleus, this green dot, as well as in the cytoplasm. And the second gene, 15245, they are only expressed in the nucleus, indicating that it is a transcription factor. So they have something to do with the, we are suspecting this as a transcription factor. They have a DNA binding protein, so they are expressed in the, they are localized inside the nucleus. So that was the status of the two genes. So one by one we'll be doing. So 11 gene we have selected for characterizing through the transgenic mean. Others students are working. And the recent result is that this 15245, apart from the salinity tolerant, it is also giving the high tolerance to the dehydration as well as ABA under seed germination. So it has a crosstalk between the dehydration and salinity as well as ABA, which is anyway well-established fact that salinity tolerance has a good relationship with the drought tolerance, their dehydration tolerance, because this is known as a chemical drought. Water is still not available for the salinity tolerant salinity stress. So the second part of this, for the bidders' point of view, in collaboration with the National Rice Research Institute, Cuttack, we try to develop the marker, because sooner or later we have to land it up to this. We have to try to develop the marker that is specific to the coarctata and also for the future breeding program purpose. So to start with this, we have taken 23,499 SSR marker, and through in silico analysis, cross transferability, we have find out that 300 marker was cross transferable to the Oryza coarctata. Out of that, 77 was the kind of useful marker, but then 2 of the markers was very specific to the Oryza coarctata, which I am going to show you now. So this is one of the marker. Upper panel is the RM11508. You can see that 18 to 23 are the different accession of the Oryza coarctata, and from 1 to 17 I have different species of the wild rice. 1 to 13 is the 5 species of the AA genome, and 14 to 17. 14 to 17, they are the primitive species under the AA genome. So you can easily see this marker can distinguish from the Oryza coarctata, which remains from the 18 to 23 length than the other wild species. Second marker, which is known as RGNM S3515, they also could distinguish the Oryza coarctata, which is 18 to 23, from the rest of the markers. So then we try to know that why there is a single band and why the band here is also single band but different length. So this work was led by Dr. Midul Chakraborty of NRRI. So we find out that the reason for this single band was that there was a 51 base pair deletion in the Oryza coarctata in this marker. So that's why although there was a single band, but this band came in the lower position. Now coming to this marker, that why there was a in panel, there was a 2 band and others has a 3 band, and Oryza coarctata has a 1 band. And we find out when you do the sequencing, we find out that there is a 2 gene in the AA genome. So in one gene, 2 gene, they have a site for this marker. And in one gene, there is a 1 forward primer, and then there is a 2 reverse primer at 2 varying length. So that's why we got 2 varying length or 2 different markers. And in another gene, which is located in chromosome number 12, there is a 1 side of forward primer, another side of the reverse primer, which yielded 1 single band, which is showing in the 3rd length. But in case of this primitive AA genome, there is no amplification in chromosome number 12 because there is no site for that during the course of evolution that has been mutated. So only the chromosome number 11, there is a 2 binding site. So that's why they got the 2 different bands. And in case of coarctata, they have a single band that is belongs to the scaffold number 572, and that's why they have got the 1 single band. So this work was published in the Euphytica, and so this was the first part of my talk. Now I'm going to the cytogenetic studies quickly. So in cytogenetics, what was known in the literature that it was tetraploid. Why we landed up this cytogenetic? The student who was doing PhD working on the crossing has failed 3 subsequent years for making any cross. Then we started thinking that there is something wrong with our approach. Then we first looked for the pollen viability. We were surprised, we find that the pollens, most of the pollens are sterile. Coarctata flower twice in a year, April and October, for a narrow window of 1 week in every places in India. So in that time, when we took the pollen from here, we did it and we acetylcarmine staining. We found that it is failing. Then we came to our mind that we must check to the cytological aspect of this. And then we find out the literature everywhere is tetraploid, but we did not get any picture or any confirmation. So we started doing the root tip cytogenetic study, and we have find out that there is a 36 chromosome. When we see the PMC, the pollen mother cell mitotic index, we found both 12 and 24. Apart from that, we have find out that 20 and 16 in number. So there is a abnormalities and clearly shows that it is a triploid. Now here there is one question. So we have made the karyogram, and our triploidy also supported by the G0 by G1 flow cytometry data. It comes in between tetraploid and diploid. But there is I just wanted to also tell you one important thing. Our work is restricted to the Sundarban. So we do not know the cytological status with the other accession which we are collecting now. But definitely it is a triploid for the Sundarban species. And when we do some other morphological study, we find out that you see the pollen of FL478 and the IR29 has a much better pollen viability than the Oryza coarctata. If you look into the seed, it produces the maximum chaffy seed and few seed-like structure. When we dehash this looks like a stone-like structure, and this is your FL478 seed, sorry, IR29 seed, and this is the original seed of the coarctata. It has a thorn in the top, and it is settling habit. Whatever seed it forms, it fall down. But none of them, there is no report that nobody could germinate this under in vivo or in vitro of this seed. So we find out that there is no embryo, and it is a seed-like, stone-like structure. Pericarp is black, but inside it is white. And we also find out, and it is also reported in the literature under stress, the growth of this plant increases. So that also give a suspension. Then we try to study the anatomy of the leaf, and we find out that this has the cranch anatomy. This has a larger bundle seed, which is surrounded by the 4 to 5 layer of mesophyll cell. When we did the electron microscope, we find out that the chloroplast and located in centrifugally, and also the grana in the bundle seed of the chloroplast is rudimentary, whereas the grana in the mesophyll cell are the functional, a typical character of the C4 anatomy. So it has a cranch anatomy, C4 anatomy. Then we did the physiological study with the help of physiology division of the IRI, and its vein density is also higher than the C3 plant. So you will find out that the carbon discrimination and carbon dioxide compensation point, does not have the functional C4 photosynthesis system. The carbon discrimination through the IRMS shows that value is minus 26.9, which comes under the range of the C3 plant. So we do not know it has the C4 anatomy, but it uses the C3 photosynthesis system. So what we can understand is that these are the 5 stages where a C3 plant evolved in the C4 plant. So coarctata has already achieved in the 3 first step. It has a higher ratio of the band density and the bundle seed to the mesophyll seed ratio. And there is a next 2 steps, so we can say confidently that it is evolving towards the C4 plant, which is also reported in many other places. When a plant grow under a very stress condition, they have a tendency, many grass family, poaceae family particularly, that they are evolved toward the C4 plant. So this paper was published also in Plant Science in a detailed study of this. Now coming to the LCMS, that many people often ask, any visitor comes to the lab, that what is there actually, what genes or what metabolites are there. So far, it was established that clinical biosynthesis pathway is the one which is responsible for salinity tolerance. But then we see, with the help of Bose Institute, we did an LCMS analysis. We found one compound, that allantoin, whose expression under salinity in the root of the coarctata is very high, higher than the pokali and the ICAR-29. So what we did is that we look into this, and the students working on this. Allantoin is basically well-known compound for the nitrogen metabolism. There is a book on allantoin. They said this is the nitrogen when the plant is a deficiency, plant use allantoin as a sole source of nitrogen. But then why it accumulates in Oryza coarctata root was our main question. So we started doing that, and then now literature is coming that allantoin is a very emerging stress stressor or stress-inducing molecule. So that makes us suspension, and then we started jumping on it. And then we have standardized the allantoin determination through the HPLC in our lab. We found out that under salinity stress, its accumulation is high in the flag leaf, inflorescence, and as well as in the root. And then the allantoin is the main biosynthetic pathway. Uricase is the major gene in the rate limiting enzyme in the allantoin biosynthesis pathway. So we clone this gene from the Oryza coarctata and put it in the Arabidopsis to see if they can bring any phenotype or not. So you see the ICAR-29 Kokalian coarctata, the allantoin concentration, and then allantoin uricase gene expression, and also we have measured the HPLC analysis, the allantoin itself, all increases under the salinity state. And coarctata, in naturally it is high. So when we put the rate limiting enzyme under Arabidopsis, we found clearly it gives a very good phenotype under the salinity stress as a salinity stress tolerance. So definitely uricase or allantoin has a positive role for salinity tolerance on this. Now coming to the RNA-seq and small RNA-seq, and we are going to detailed study on this. We have also find out that allantoin induced the bacillus-steroid pathway, and then the whole system grows, and it induces the lignification, cellulose deposition in the root. So those kind of work is going on. It is not finished. But then what I want to tell you that allantoin is definitely one of the important metabolites that take important role for salinity tolerance in the Oryza coarctata. Coming to the RNA-seq study, so we did first the small RNA library from here, and then we also made the degradome library from this Oryza coarctata, and we reported all kind of microRNA data present in the Oryza coarctata, and the salt-induced this. But while working in this, one of my students has a bioinformatics background has pointed out 2 important things which I would like to share you very shortly. One of the things is that when we measure the microRNA stuff for any plant, people report normally it should be exactly matched with the mRNA, because the function of the microRNA is to degrade the protein coding gene. The microRNA cut the protein coding gene, so the protein synthesis is being stopped. So we do the thing, identify the microRNA and their target, that is the mRNA, by matching the some seed region. So ideally it should be the green in color, but there is a varying type of approach as taken in the literature. It allows the mismatch from 2 to 4. Later on we reported that that is not correct. We should look for the 100% match, then the data is genuine. And then that student has also find out from the same set of data, one of the widely used software for determining the microRNA is the MIR-DEEP-P2. So people, what is they used to do, including myself, when I published this paper, we ran the MIR-DEEP-P2 only once to identify the known as well as novel microRNA from the small RNA-seq data. But we have also find out now that at least you should repeat the using the same data set, because in every run, if you repeat the mid, deep, P2, we find out the red color top portion, 10 to 20% microRNA, every run is repeated. If you do not run and if you publish the paper with a single run, then many information from the same set of data we miss. So by doing this, we have demonstrated that we can dig out many more new information on the microRNA from the microRNA sequencing library. Here is an example. Another important point we find out that when we publish this microRNA result, we have used the Oryza sativa genome as a reference genome to identify the known microRNA. The reason was there that time the genome sequences was not there. I was in NBPGR, so we don't have any other alternative. So then we use the Oryza sativa as a genome, as a reference genome. With that, we identified 52 known and 87 novel. But the same data set, when you use the Oryza coarctata genome, you see the incremental increase of the microRNA discovery. So what we recommend is that if you are working on the microRNA, please use the same species reference genome. Otherwise, you will miss many. And some of these microRNA here are Oryza sativa specific. Some of the microRNA are Oryza coarctata specific. Some of them are common. So to validate that, we have chosen randomly 5 Oryza sativa specific microRNA. And then we have taken the Oryza sativa genomic DNA, Oryza coarctata genomic DNA to amplify those 5 Oryza sativa specific microRNA. You see they have amplified against the background of the Oryza sativa. We have also chosen the Oryza coarctata specific microRNA that has amplified when you use the Oryza coarctata DNA and did not amplify in the Oryza sativa. When you choose the 5 random common microRNA, they amplify both. It shows that whatever we have identified through the bioinformatics is exactly matching. And I'm very happy that this work was published in a journal which has recently got an impact factor of 14. So entire pipeline is there. Then we have tested our pipeline with some published data. I'll very briefly show you in the chickpea. This is not a coarctata specific pipeline. Now one can use for any plant species. So there was a published paper on chickpea. There was a 2 genotype they have taken. You can see the microRNA predicted with our pipeline using the same parameter 56, 32, and non-redundant is 70. Between the from the 2 samples, we have used the same parameter which was reported in the paper. We have used the same length of the microRNA what was reported in the paper. In the paper, it was reported 31, 13, and 38. You can see the difference with our pipeline. It has increased to 56, 31. Please remember, we are using the 100% match. We do not allow any mismatch. Using that, we have taken out this using the chickpea genome, reference genome as a sequence. What I want to say that if you use our pipeline, then your yield of microRNA from the same data will give the much more than the existing pipeline. So this was one of the things, and this is, as I told, that we have it's a failure story. We have made up lot of breeding effort. Now we are thinking that we'll be using Oryza coarctata as a female parent. We'll not take the pollen from here. We'll be taking the pollen from the rice, and then we'll be dusting on the Oryza coarctata. Let's see how it works. All the work, whatever we have done. So in conclusion, that genome size was determined around 665, and this was the first monopod halophyte which has been sequenced and assembled. And then chloroplast mitochondrial genome was decoded. We are identified at present 11. Now I have got another DBT funded project where the project itself is discovering the high throughput gene from the Oryza coarctata. So we are expecting we'll be discovering many more number of genes, but this 11 was from the genome sequence data. So it is under process of the adopting the C4 plant. We have find out that this is triploid, but our work is restricted to the Sundarban collection. Need to do the wider samples which you are planning. It has a cranch anatomy. Several salinity responsive microRNA has been discovered. Allantoin definitely play a major metabolites or one of the major metabolites which play the salinity adaptation. Repeat content is the lowest among the 27 genotypes. 27 species among the genus Oryza, but biological significance is not yet known. This is most primitive, belongs to the tertiary gene fold, and also it is known as amphibian plant because earlier people used to think that this was aquatic. Now it becomes terrestrial. It grows very well as a floating rice for a long time. So that is the most primitive species for this. The future strategies, what we are taking is that now we are doing collaboration with one premium institute. We'll be collecting the different accession and we'll try to find out the different variability of the salinity response. If it find out, then we'll take the association approach and then identify the gene, validate in Arabidopsis, then we'll go for the transgenic way. Because until unless I'm not very confident to say now that we can make a cross with this to the rice. Another approach which is very new, and some of you may be knowing, the new domestication. If we can do the domesticated de novo of this Oryza coarctata, then we do not need cross with any rice. We can make the Oryza coarctata converted to a rice. For that reason, we are standardizing the regeneration protocol. One PhD student is working on this, and then we'll follow the genome editing. We'll be edited the domesticated gene and try to make it as a cultivable or commercially viable plant. And the third one is that it will be trying crossing with as a female parent to transfer the gene of the rice. So I have finished my talk, and whatever I have told, this is already there in the database form, so anybody can use this. We have assigned all the SSR in the scaffold, and we have the total number of genes. People are using this. At least 3 institutes are taking data from us, and we'll be happy to give. It is already in the public domain, and one can download the chloroplast and mitochondrial genome, and that has been mounted to the NCBI. That is the thing. So with this, I have finished my talk, and then we have come up with a book on wild rice on which the coarctata is written there. And I must give a thanks to the person concerned who encouraged me to do that. Dr. N.K. Singh is a very, all of you know that is a very living legend for this genome sequencing work. Dr. Tia Sharma encouraged me heavily when I joined here in this institute to do this thing. Our present and previous director and my collaborator, Dr. Rakesh Pandey from the plant physiology and Midul Chakraborty from the NRRI. We are also having plant with the Midul. And one farmer's friend who gave me given a lot of information and knows this coarctata plant. Is a very aged person. He knows very well, and he is located in the Sundarban area of 24 Ganas. And this is my lab student. The Sony is the main person. All paper is Chaurasia et al. He has recently finished the PhD on coarctata and joining shortly to the Banasthali University. He has got a job. 2 other students are doing work in the coarctata and their PhD work. Hukum is the main person for all kind of bioinformatics, Hukum Rahul. And Savannah, and then S7 scientist, has joined in our group and working on this. So with this, I finish my talk. Thank you very much for your patience hearing. Thank you, sir.
Thank you, sir. Thank you for an excellent presentation on utilization of Oryza coarctata and its genomic studies and several aspects you have covered. And now the floor is open for the discussion. If any question from the audience, we will take up.
First of all, I would like to appreciate the excellent talk given by Dr. Tapan Kumar Mandal. Sir, you have presented a wholesome dish, not only covering 1 or 2 facets of this important wild species. You have covered all aspects related to research on Oryza coarctata, which was earlier known as Potricia coarctata. And we are indeed delighted to hear the good information what you had presented. Sir, I have 2 queries or suggestions. In the evolutionary history of different wild species of rice, where exactly like Oryza coarctata fit in? Because that is one information I couldn't find in most of the classical textbooks. And when exactly, like say, the species diverged, and from, if it has diverged, from which other wild species it has diverged?
It's a very good question, and it's very difficult to answer this. But what we could do, there is a software and there is a standard analysis for the evolutionary clock of any plant among the whole plant kingdom. Here we find out that it has the 10 million years ago, it has been separated from the other species of Oryza. But more than that, I cannot tell you. But one of the important thing is that the repeat sequence data from the from our lab as well as Professor Rode Wing. It is coming up now. He's also made the genome sequence of the Oryza coarctata. It is also under consideration. It will be published. So this is the most primitive. It will be placed in the top of the Oryza species. But as you said that from which plant species it has been diverted, is yet to find out. The main problem is that if you do not have the very well annotated reference genome, so till the time you cannot answer this, because our genome sequence is a draft genome, there is a lot of gaps. So we are doing now this. So because of that reason, we are improving our genome sequencing. So once we get the chromosome level platinum standard genome sequencing, definitely we'll get the answer of this. But as on today, we only know that it has been separated from the AA genome 10 million years ago, 10 MYA.
Sir, one more my query or comment is regarding the future approaches to study and then exploit the genomic variability in Portricia. What exactly is the level of diversity, at least based on morphological traits among different accessions of Portricia or Oryza coarctata?
That itself is a project now. We are recently got the funding, collecting the, because as I told in beginning my slide that nobody is having the germplasm collection. In NBPGR Center of Kotok, they have the collection from the Vitorkonica accession. So it is very difficult to answer it now. And we try to contact with the foreign worker working on this. But until unless we get the different accession from the different region of the coastal region of the India, we cannot tell you. So that is what we are going to do.
Because I see one major bug in it, because for association mapping, you need enough diversity, and the population also need to consider a population with a good structure. That's what I'm a bit worried about. And as far as breeding work is concerned, we now have a pioneer who worked for many years. Even his PhD work was on Portricia or Oryza coarctata. I would suggest you to kindly have a discussion with Dr. K.K. Jena, who is working as professor and head School of Biotechnology at Kalinga Institute of Information Technology. Probably, in addition to the approach what you are adopting, in terms of how to exploit or develop introgression lines, I've seen a couple of his publications wherein he has developed some introgression lines. Maybe he'll be able to show further light in terms of how to exploit the important traits through breeding. It's my humble submission, sir.
Yeah, sure. Because Dr. K.K. Jena is one of my mentor of this work, and he visited twice to our lab, and he knows my work very thoroughly. So problem is that first of all, we are planning to collect at least 100 accession from the Gujarat coast to the Andaman area, but it is fast bewildering. It is vanishing very quickly. So we are making now an approach. And if anybody is interested to join their hand, it's most welcome. Once we get the collection, then we'll go. We have a plan to go for the resequencing, because we'll have the now genome sequencing data to not only know the genomic level variation as well as the morphological variation that exists in the native place, and also we'll be getting. But if we get the sufficient variation, then only the association panel will be useful, as we said. But then somebody has to try, and we have got a project, we'll be doing this thing. Second thing is that Dr. Jena and Dr. Darshan Singh Brad, both who is not here now, they came number of times. I was very much, how to say, I discussed time to time with them. But both of them, they said that they could produce some F1, but then further they could not produce. And that too, F1, they have produced after a lot of try. Like Dr. Jena, he attempted 20,000 crossing, and after that he got one. But then he now also I am in touch with him. He's in Kalinga Institute of Technology, Bhubaneswar, and he has some technique with, covering with a black cloth. So let's see how it there. I am very much well connected with him for this particular work. He visited twice and saw the plant, and we discussed a lot of time. But then breeding step is very difficult to nobody, not only from India, but no population, nothing is
Sir, one last question or query. What is encouraging is some of the microsatellites or SSRs are transferable across coarctata and Oryza sativa. Did you try the same thing with the commercially available SNP chips like 7K chip or even our own indigenous 51K chip? Probably, like say, if some of the SNPs do show some amplification across coarctata species, we need not even resequence. We can use the presently available chips for association mapping, sir.
Let us collect this. Very good suggestion. What you told that we have discussed this. Even the simple SSR also will give us we know the polymorphic SSR in the now, but then we do not have any live germplasm collection. That is hindering our work. Neither not NVPGR, nothing. So
as a beginning, what you can do is the 55K chip is your own chip that belongs to NIPB. With that, you can try, say, we have an accession of Portricia coarctata with 2-3 accessions of Oryza sativa. You can try it and then see if those chips do show some polymorphism between sativa and then coarctata. That is something which I would strongly suggest.
Correct. We'll be doing that.
It was a wonderful talk, sir. Thank you very much for obliging our request and then delivering a talk. Thank you, sir. Others, sir. Yeah.
So can you unshare your screen so that we can go for further discussions? I request Dr. Mondal sir to unshare his screen. Yeah.
Sir, thank you very much, sir, for your wonderful talk. This is Krishnamurthy from CSSRA. Sir, in your last slide, you are telling Oryza coarctata should be used as a female. In our breeding program, usually all land races are using as a male to avoid the genotypic cytoplasm effect, undesirable effect. So what may be the reason Oryza coarctata should be used as a female, and how to interact as the gene to rise from Oryza coarctata?
Yeah, that this is a suggestion from the breeders only, because it does not have much pollen viability. That is one of the reasons why the breeding work is slow. So they say that when the cytological abnormality as a female is much more lower than if you use this as a male. So if we make any cross or get some seed from the coarctata itself, those F1 can later on be used, changing the breeding strategies to transfer the rice. But anyway, that's the portion which is looking by the Dr. Gopalakrishnan from the IRI division with the consultation of the KK Vinod. So as per their suggestion, we plan this year, because using this as a male will have I can definitely say that it will have a very low rate of success, because it's diploid in nature. What we came to know now, people are thinking it is a tetraploid. And then second thing is that the pollens are mostly sterile. That's the reason.
Is only because of the unavailability of the pollens. That's why you are using as a female.
That is the main reason.
Yeah, Dr. Krishnamurthy even faced the same problem with Oryza longistaminate. I personally worked with that, and Dr. Girish will also support the approach of Dr. Mondal. Am I right, Dr. Girish?
Yes, sir.
Sir, what exactly we are following, sir? Because all white races are not good agronomical background. That's why everybody are using as a male instead of female. If you are using as a female, there will be a more chances of getting the undesirable character in F1s or in segregating population.
See,
that's why I'm asking.
Yeah, you are right. But the fact is that, once you develop the integration lines, whether you use this as a female parent, and then the derived lines can be probably used as a donor. But that time you can rectify the cytoplasmic issues. So initially, to begin the integration of the genes from the Oryza coarctata into a sativa, probably the approach is very much correct, because the pollens are not fertile.
Okay.
So
Tapan, sir,
once you develop the pre-breeding material, you can go ahead.
What may be the reason why the pollens are not available in Oryza coarctata?
That we need to look into it. Nothing, not much is known. As I told you that, first of all, it flowers for a week. And within all plant will be yellow in color because of their pollen, and then it will, they die. and there is a very few seed are formed seed-like structure. But since it is a triploid, so we suspect that it is widely reported in the triploid. It is a typical character of the triploidy that the pollen and the lace set of the seed, or the producing the chaffy seed, or producing the few seed. It is widely reported in the literature.
Sir, have you tried the Oryza coarctata into Oryza coarctata? What will be the success rate of seed setting?
We have discussed, but knowing that it is a triploid, so it will be now difficult to do this thing. And then we have not tried that, because even if we got it, until unless there is a kind of variation between the two parents, so then that is what we are looking now. That will be collecting the.
I think that will give us a solution to why.
Yeah, because we have only one collection, so that's why we have not tried. And emphasis on our work is not on the breeding basically. That's why we are collaborating with the Dr. K.K. Jena and Dr. Gopal is there. But nevertheless, one day or at the end of the day, we have to transfer the gene from here to the rice, either through the transgenic approach, or through the conventional breeding, or through the de novo domestication. These three of the things. We will look into it. Until unless we are not getting two contrast parent of the Oryza coarctata in terms of anything like salinity adaptation or some morphological thing, we are handicapped to do that.
Sir, can I ask one question, sir?
Yes, sure.
Yeah, please, sir. Please.
Yeah, sir. I have been collecting a Coarctata coarctata, now Oryza coarctata, since last 20-30 years. If you see, sir, in Bhitarkanika area in Oryza, you find miles and miles in mangrove forest. This Oryza coarctata, exclusive, all species available with Myristicus, that one Wheatiania is there. That is the association species with Coarctata. But then if you are telling that it flowers in April and October, but I observed in Bhitarkanika, it flowers in February and August. And that is maybe the location specific, maybe location.
Yeah, what we came to know that in the in Arizona, it flowers in throughout the year under the control condition. Basically, it requires 30° ±2 for flowering. So when the temperature comes there, if you see that in Delhi, April beginning and October, the temperature comes around 30. So that time it flowers. So the change of the place may be related to the optimization of the temperature, which may induce the flowering.
One more, yeah. Sir, one more observation. I had gone to Andaman 2-3 years back, and I didn't find a single occurrence of a Coarctata and Oryza coarctata over there.
Very important thing you have told. I just wanted to have one minute this thing. Now the thing is that if you do whatever, you need the number of accession. So to collect the number of accession, I have contacted many people, many research institutes in the coastal region. What I came to know is that I told in my lecture also, it is quickly vanishing, and it is not available due to the tidal force or whatever the reason. It is grazed by the animal. So it is the place where the coarctata was available previously. Now it is not there. So it is a high time to conserve that. But now I have got a information where in the Andaman it is available. It will not be available in the tourist place, or it will not be available at place which is has a high human activity, like in the Sagar Island of 24 Parganas. It was a full with the coarctata, but now the senior people say that it was earlier, but not now. So we have now concentrating, and we have now has the information where it find out in the Andaman, and it is still there in Andaman with the with association in some mangrove area where the human activity or the tourists are not restricted to go there. That is what not only the Andaman, and what you told is very correct. It is every places. So 7 places we could locate at present where the coarctata is available in the Indian coastal zone.
Sir, in fact, I had gone there to collect this Indandamanica species, or as a Indandamanica. In quest of that, I had gone to several places in Andaman. No, I didn't go to Nicobar Island, but only Andaman, Port Blair, and other regions I have visited. But I never found a single plant of Coarctata coarctata. And sir, one more thing before you close, sir, I just want to add to. If you come to Bhitarkanika area, please traverse through Dhamra Port, and you'll find miles and miles of Coarctata. not a one or two population, and continuity of Potracia. So yeah, sir, and that too, one more suggestion I can give, sir. You forget about flowering time. You can come anytime. One can explore and collect the different accessions. That is what my suggestion is, sir.
Thank you very much. Maybe I'll be contacting with you. I'm in a touch with Dr. Pani, so it is good that I'll be in touch with you. This thing. Even in the Sundarban area, Sagar Island, Bokhali area, it is goes like I shown in my first slide that one cow is standing. It goes like a bed.
Yeah, I have several photographs, sir. If you want, I can give you. And more, sir, one more addition I wanted to tell to the gathering here. Sir, the meaning of this coarctata is the seed is compressed and it has a lateral wings. And so that when it sets from the plant, it can float and settle elsewhere. That is why coarctata. It's seeds. Yeah, that's what. Thank you.
Sir, I'm having one question. Yeah. Can I ask, sir?
Yeah, please.
Myself, Ananta, sir, working as a scientist in plant breeding IIRR, sir. So it's a really excellent talk, and earlier also I read a few of your important publications. It is really so very much excited. So in your presentation you mentioned that allantoin is the gene or this metabolite responsible for salinity tolerance in coarctata, and you are transferred to Arabidopsis and found that it is effective there also. So is there any effort to transfer to cultivated Oryza species and tested, sir?
No, we do not have any plan on that because, you know, we don't have the manpower and resources to transfer or to make the uric acid to the rice, and there is no point because these genes is also present in the rice. So when we do the transgenic work in rice, I make sure that there should be some purpose. Either genes should not be present in the rice genome, or there should be some other reason for functional validation of this. So our main interest was allantoin accumulates heavily in the root in the coarctata plant under salinity condition, like pinital biosynthesis pathway. So uric acid is one of the rate limiting enzyme in the biosynthesis of the allantoin. So when we take this from the Oryza coarctata and put it in the Arabidopsis, it shows clear standard salinity tolerance. So that confirms that this has some relation. Now with the RNA-seq data as well as other HPLC analysis, we find out that it induces the bacillus steroid pathway gene. And then bacillus steroid does the further downstream processing. Along with that, it also induces the lignification, cellulose, ROS, etc., which gives the tolerance to the plant. We are interested to find out the mechanism mediated by allantoin biosynthesis pathway for salinity, but we don't have any intention to make the transgenic rice using the uric acid gene because that will not have any impact. Already rice gene pool itself, there are many tolerant gene or alleles are there for salinity.
Thank you, sir.
Sir, I have a question. When you talk about such a metabolite biosynthetic pathway like allantoin biosynthetic pathway, these are all energy intensive pathways. So if we try to transfer this particular trait through any means into cultivated species, there will also be a metabolic cost because if you look into Oryza coarctata, as far as flowering is concerned, in my early days, I used to observe a few coarctata accessions which were available here, and their flowering is very, very sparse even during peak season, and the seeds are almost very shriveled or malformed. And is it because the plant is spending a lot of energy in terms of tolerating the salinity, heavy salinity stress, or is it because it's a natural adaptation?
I don't think that salinity, maybe the natural adaptation is this thing, because I do not know that why this flowering is so short. That aspect, actually, I have a lot of work with the coarctata, so that aspect, this is all are generating now. So we did not find out this thing.
Now, my apprehension is, of course, I hope I'm proved wrong. We may find excellent tolerant accession, but when we try to transfer this trait into cultivated, it may also involve a lot of energy wherein the plant wastes a lot of energy. We observed the same thing even with transgenic rice, which had TPSP, in the sense that transferring TP or Trehalose biosynthetic pathway genes, they conferred excellent salinity and drought tolerance. But the plants did not show the same kind of agronomic performance when compared with normal non-transgenic plants of the same variety. It could be just this thing only, apprehension. I hope I'm wrong, sir.
Yeah, that needs to be looked into.
Dr. Lothant?
Sir, I'm Dr. L.K. Bose, working on wild hybridization program. Last 10 to 15 years, I'm specially working on wild hybridization program. Sir, you have given excellent talk, really excellent talk, sir. And in my net house, I am maintaining 3 to 4 accessions of this Oryza coarctata. Okay. I have crossed with different genomes, starting from granulata, CCDD, tetraploids, glandulumis. And also I tried with this Oryza coarctata. So the cultivars which I have taken as a female and coarctata as a male, okay, I tried 10,000 crosses and what I've seen. And in most of the crosses, settling is there, and on 3rd day or 4th day of emasculation, a pollination, the pollen to pollination is there, but after that abortion of embryo starts. So I did all type of gibberellic acid, IA, ABA spraying. So settling was stopped. Later on, what I got information that instead of taking diploid sativa, we have to take tetraploid sativa. So you will get a success. So even you take also female as a cultivar and the male as a coarctata, but we have to follow, because in most of the latifolia and the tetraploid, glandulumis and minuta. So in that cases, I got number of lines. And when I'm using tetraploids as a cultivar or cultivated rice by cultivation treatment, then success are okay. That's all.
I will have the comment on that. Anyway, that breeding aspect has to be taken by the breeder. It's neither a mandate of the NIPB, but I want that there should be some breeding success so that whatever the genome sequence and other work I am doing should go to the field of the farmer for the benefit of that. But I have a well connection with the Dr. Jiva Siraj of the Dhaka University. She also came here. She knows my work details. She also did the crossing with a tetraploid rice and the coarctata, and has got a limited success again. After getting some seed, when we asked to for collaboration for the through MTA and other thing, she could not give this thing. So what I want to say that if really if anybody can come up with some crossing with this, that will be excellent. You keep trying, and I'll be support from. I'll give the support the other thing like the markers or the genes, what is present there, or the metabolites, what is there, which is the domain area of my own research. And I'll be complementary to your work if you can come up with whatever the means, whether it is a tetraploid as a rice or diploid rice. But I am quite confident the Sundarban collection is the diploid. Why I'm telling is that when we look back to the literature, I did not find any body is having any photograph of the tetraploid. And I particularly suspect one of the reasons for less progress in the breeding work is that it is a triploid in nature. And if you take the triploid plant crossing with a diploid or with a tetraploid, naturally you will be getting a jerk. As you are saying that you have tried 10,000 cross and did not come up with a one thing, and after the embryo abortion and others. So this is one of the points. Second point, I have talked to almost all the people who are living on anywhere in the world on the coarctata. None has the experience to germinate them even in vitro at the mature seed. It's a highly recalcitrant seed. Only the immature seed they could germinate, even in the Bose Institute, Swaminathan Research Foundation, Dr. Ajay Parida, and then many other people. So this all shows that something wrong with this in the cytogenetics level. So that's why we find out. Now I have to look for the other accession, whether there is a tetraploid, it still exists or not. So that is what we are doing now. And if we find out, then we'll be making some, we'll tell the breeder to take up some program based on that. but handling with the triploid plant for the breeding program will be difficult to get across. Even if you get some by chance 1 or 2, will be difficult. But anyway, it is your domain, so you can keep trying, and I'll be helping if somebody needs any genomics information from here, that I'll be giving. And if anybody should not overlook the C4 machinery which is having there, because Bill Gates Foundation has spent millions and millions dollars to make the rice plant for the C4. So here there is a natural source of the C4. If it belong to the genus Oryza, so if also some gene or something can be transferred, that is also will be the wonderful.
Thank you, sir.
Thank you, sir. I think, are there any questions from the house?
But lastly,
if there are no,
I love to collaborate with anybody. If anybody, this thing, particularly breeding aspect or plant physiology, we have all other data also. I did not publish that how it manages the sodium potassium ratio. What is the salt gland? We are also trying to identify the salt gland which is present there and try to make the RNA-seq from there to identify the salt gland specific promoter. All those basic academic works is there, but from the both the Rice Research Institute, even from the kernel, if some, even I have a contact with the regional center of kernel located in the Gujarat. So for this plant, so if anybody wants to make any collaboration, then I'll be very happy to collaborate. Our director will also be very happy for this.
Yeah, sure. Yes, sir. Thank you.
Thank you.
So there are some queries in the chat box, sir. Maybe we'll try to read out.
If you have a time, I can see this thing quickly.
Yes, sir.
And,
Yeah.
okay, just,
I'll read, sir. I'll read for you.
Bank session.
Yes. So are there any plans for exploitation of genes for biotic stresses? From the data available, any specific defense pathways are present in this? This is the query raised by AP Padma Kumari.
No, so far nobody has done, and that was the main point of attraction of the Dr. T.R. Sharma, who encouraged me when he see that it is not even the blast, he is a man of blast, this thing. But there is no disease and pest. I have not seen from the 2014, and I told you that person who gave me the plant, it is very, this thing. There is no, because leaves are so leathery and thick that it is difficult for, that is one of the reason, could be. So there is no
possibly one of the reason, as I understand, I also tried in the initial phase of my career, tried to inoculate bacterial blight pathogen on Potricia. It shows immune reaction. It also means that probably it's a non-host for many of the pests and pathogens. Being a non-host could be one of the reasons, sir.
Maybe that is the thing. But there is no report for any pest and disease for this.
So there is one. Yeah, there is one more query from Chitra Shankar Madam. What is the pest spectrum in wild?
Pest, I did not understand. What is that?
What is the pest spectrum in the wild?
What she wants to know is there any pest which naturally occur in the wild rice collection?
That information I told you, those who knows this, those the fishermen and the farmers of the Sundarban who is having this plant nearby their house. No scientific data, first of all. In wild condition, I asked them number of times. Nobody has seen any this thing, any pest disease for this.
Yeah, so there is one more query from AP Padma Kumari, madam. What is the soil condition do you need to maintain the species in the institute? Do we have to create a mangrove-like situation, or is it feasible to maintain on the normal size?
Size means spacing you are asking?
Soils.
So, sir, in terms of like, so how much easy you have to maintain in the pot? Probably.
Yeah,
oh, okay. So I show you, you put one plant, and then if you give the under net house condition, it requires some natural, it's like salt we give like a urea. If there's a distinct difference of the morphology if you do not give the salt and if you give the salt. So we apply the 200 millimolar salt intermittently, and in one season the whole pot become a bush. So it grows very fast, and the biomass production is also very high, higher than it is well reported, and the Rubisco enzyme activity is also higher than other species of the this thing. It is reported from the Bose Institute and IRRI also. So it grows very fast, and one pot, one plant sapling we put, and in a year end it becomes the food.
Sir, are the leaves edible for feeding by cattle, sir?
Yes.
Or okay.
Yes, the cow and buffalo, they feed the leaf very nicely. Although the leaves are serrated, having some kind of rough thing, and seeds are very dangerous. If you touch it by chance, if it comes, it is a very good thorny material. Definitely, your if you're not careful, the blood will come from that thorn. But the green leaf is well eaten by the cows and buffalo.
Yeah, there is one suggestion or point from Guru Raj Katti sir. As per the Dr. Patra large area available under this species, pest data may be collected from there for a realistic assessment.
Yeah, that we are, myself and Dr. N.K. Singh will be going this
Yes.
November to various places.
Yes.
Collect this. Very much
I
that,
think-
other
Yes,
places.
I think that's all about the question, I believe.
Yeah.
So we can go to.
Before Dr. Neeraja offers her concluding remarks, it's my duty, sir, on behalf of our society, Society for Advancement of Rice Research, we would like to, I'll just remove my background, sir, just one.
Yeah.
So we would like to present you a memento in honor of your good self for delivering a very nice talk. Thank you very much, sir. Thank you. I'll pass it on when I'll be coming to Delhi. It's a memento from our side for your kind acceptance for delivering the talk, sir. Thank you.
And I was little scared because I just recovered 2 days before. Dr. Girish knows. So
Yes.
I was not coming to the office for past 10 days. But it was so tempting to me that an invitation from you and from ICAR-IIRR. So I immediately accepted this. Otherwise, due to some reason, I was not well. I was not coming.
After you get well soon, your physical visit is also pending. You need to come here. There are a lot of good youngsters in both breeding and biotechnology. And then we invite you to have a discussion and then motivate them for in the frontline areas of research, sir.
Thank you.
Thank you.
Sir,
Girish, we
thank
can
you, sir.
move.
Yeah. Sir, now I request Dr. C.N. Neeraja madam for her concluding remark.
Thank you, Girish. Dr. Mondal, it is indeed a very meticulous scientific study, I can say, because you have collected the species and you have gone up to the characterization in terms of their chromosomes, their genomes, and the genome, genes, functional annotation, transcriptome, miRNAs, and everything. So it is as from science perspective, it is a very meticulously studied species, I would say. So, and we look forward for your association in CRP biofortification also. Sir, Director, sir, we have he has been proposed as a partner in the new plan scheme, and and especially there were around 75 to 100 participants at one time. So we can that shows your reputation as well as the interest on the topic of the wild species. So and the response it has elicited from various participants, including our director and also the other staff. So we thank you very much. I think formal vote of thanks will be proposed by Padma Kumari madam.
Thank you, madam.
So can I go ahead, sir?
Yeah, go ahead, ma'am.
Thank you, sir. So all nice things should come to end at one point of time. So it's my proud privilege to propose vote of thanks for this webinar. On behalf of ICAR-IIRR, SAR, and my personal behalf, I earnestly thank our director for coming up with this idea to take up this webinar, and Dr. Tapan Kumar Mondal, Principal Scientist, National Institute of Plant Biotechnology, for accepting our invitation. Literally, this webinar has been postponing for so many days for want of suitable dates and time. And finally, we could do it, sir, despite many odds. Thank you very much for obliging our invitation and delivering this talk. on this. This is the 14th webinar in the series as part of the Azad Ka Amrit Mahotsav celebrations. We are very much thankful to you, sir, for sharing your knowledge related to this unique species. Now we have genome sequence for 2 Oryza species, and it's congratulations for being for sequencing this Oryza genome. And this is a platform where you have shared your knowledge, opening new avenues for research and collaboration and for future deliberations. Now, I also thank all my colleagues who were part of organizing this webinar, my seminar colleagues. Then Dr. Padmavati, Dr. Neeraja, Dr. Divya, Kalyani, and Dr. Sailaja, head AKMU team who are instrumental in conducting this webinar. I also thank all the dignitaries, Dr. Padmini Swenmem, Dr. Director NRRI, then Dr. Patra, Dr. LK Bose, who have actively participated and shared their views, and all the retired scientists, members of the SAR, scientific fraternity, ECRIP collaborators, and other RISE fraternities from sister institutes of ICR for actively participating and coming up with good comments, suggestions, ensuring fruitful discussions. I also thank Dr. Girish for moderating this session and Dr. Divya and Dr. Shruti for recording the proceedings. Thank you, one and all. Thank you.
Thank you.
Thank you very much, sir. Thank you very much. Thank you, everyone. So we'll close the meeting, ma'am. Thank you.
Yes, sir.
Thank you, sir. Thank you, sir.